Welcome, everyone. My name is Chris Callos for MST Access. We're joined today by James Graham, CEO of Recce Pharmaceuticals. Recce Pharmaceuticals is a clinical stage biopharmaceutical company developing a new class of synthetic anti-infectives, which hopefully will address antibiotic-resistant bacteria, commonly known as superbugs, and viral pathogens. The lead candidate is Recce 327, which is in a variety of clinical programs. And with that, I'll turn it over to James to walk us through. Thanks, James.
Hello, Chris. Good to be with you. And thank you, MST Access, for this opportunity to present Recce Pharmaceuticals. Today, I have a usual investor presentation I'll bring up before us now and just sharing on screen. So, Recce Pharmaceuticals, usual disclaimer. Recce Pharmaceuticals is, according to World Health Organization and Pew Charitable Trusts, clinically the most advanced new class of antibiotics in the world at this time. It is the most advanced sepsis drug candidate, sepsis, the technical term for bacterial blood poisoning. It is the only qualified infectious disease designated candidate for bacteremia, which is again a technical term for sepsis, by the US FDA. So that qualified infectious disease designation provides us 10 years of market exclusivity above and beyond our patent position, plus expedited regulatory review for the life of the regulatory process. This synthetic class of antibiotics is the most advanced synthetic class. All existing antibiotics are naturally derived, so they have expensive waste, they have high capex in manufacture, and unlike our compound, they don't continue to work with repeated use. We work as well the first time, third time, fourth time, all the way through to many, many, many repeats, which means we're valuable today and will continue to be valuable in the future. Recce has multiple clinical candidates, and we share a number of them beyond. So briefly, who am I? I'm James Graham. I'm CEO and Managing Director. I started as the founding investor, in fact, quite honestly, supporting my grandfather. My grandfather, Dr. Graham Melrose, was former head of Johnson & Johnson Research Australasia, which is, of course, a third of the globe responsibilities. He had that role for about 10 years, as well as executive director of their board. And he really looked to the world differently, of course, looking at the challenge in the medical setting and putting on that chemistry hat to design a compound that works against the characteristics of antimicrobial resistance. So I joined as a passive investor, supporting my grandfather getting out of his garage into a proper lab in around 2012. But really, I joined actually as an employee, which wasn't the intention, but I did in about 2015, executive director of the board for some 5 years and CEO for approximately the last 2 years. Surrounding me is fellows like Dr. John Prendergast, our executive chairman. Dr. Prendergast has been with us for some 6 or 7 years. He's chairman of a New York Stock Exchange biotech company, lead director of a NASDAQ biotech company, and is really got a doctorate in the study of bacteria and blood diseases. Dr. Alan Dunton is our chief medical advisor. Dr. Dunton, in the bottom left corner there we see, is former global head of Johnson & Johnson Research, known as Janssen Research. He actually has gone from concept to commercialization with 3 previous antibiotics. I think there's only ever been about 20 created, and 3 of them were under his guidance, and he leads our medical programs, as well as, of course, many other colleagues. So Recce, as a capital snapshot overview, we're valued at around 100 million Australian dollars. Our cash at bank we see there of $8.52 million is actually before a placement, which isn't on the screen at the moment, but we did a placement last week where we received an additional $8 million from institutional investors, and we have an open SPP for $2 million from existing shareholders. That gives a pro forma cash position of around $18.5 million, of which then actually we received a US Department of Defence grant only a couple of days ago of approximately 3 million Australian dollars. And I think we touch upon that in here. So we're very well funded. We obviously received the Australian R&D rebate. Uniquely in our company's instance, We're one of the only companies, you can pretty much count them on one hand, that has two things. One, that R&D rebate extended anywhere around the world. So right now I'm actually in Indonesia, by example, and that 43.5 cents R&D rebate is extended across to our activities here. And we'll talk a little more about why I'm in Indonesia at this moment. It further guarantees up to $55 million of expenditure that if we do submit R&D, no matter if legislation changes, government changes, all the usual stuff, they guarantee that they will repay us up to a quantum of $55 million. So there you've got non-dilutionary cash protection and a significant cash offering should we go to claim such. Oh, we did have it on, yeah, right hand side there, 8 million raised. So well done, marketing, thank you. Why are we different? So all existing antibiotics to date are naturally derived. They're bacterial or fungi, they're cultivated out, and they're put against the offending bacteria or fungi as an antibiotic they wish to kill. The challenge is you have really much like a lock and a key. If one was to say there's the antibiotic and then there's the bacteria. The two come together today in a harmonious fit and the antibiotic wins. But the bacteria seek to survive. They always have, they always will, and they don't like to lose that fight. So they mutate, they change their structure, and the antibiotics are not overly smart. They have fixed structures. They're only as good as what was given to them in nature decades ago, and that lock and key mechanism no longer fits. The other challenge, of course, of existing antibiotics is they're generally very specific, not only in their bacterial fit, but in their bacterial efficacy. Only certain types of bacteria will they work against. And they come with a lot of toxicities as a result of being found in nature, not designed from first principles. Recce is designed from first principles. We've created what's called a polymer. So it's a small beaded molecule of many components stuck together with desired characteristics, not just by what was given to us in nature. And that means that we're attracted to the proteinaceous layer of bacterial cells. We bind to that bacterial cell, regardless if it's a gram-positive, if it's a gram-negative, or if it's a hypercellular mutated form, or got biofilms, or all sorts of other things they throw up to try to protect themselves. We permeabilize that cell wall, and we shut down the bacteria faster than any other antibiotic to date, and we shut it down non-specifically, meaning it doesn't matter the bacterial stains, and it doesn't matter that hypermutated form. Of course, that therefore means that we've designed our compound to be capable in the blood all the way through to the opposite pH acidity of the stomach. And that allows us to have multiple formulations, IV, topical, so in a gel or a spray onto the surface of skin, aerosols, intranasal, so inhaled into lungs, sprayed into the sinuses. It basically is positioned to, as long as it can get access to the bacterial infection, it works and it keeps on working with repeated use, the fundamental challenge of all existing antibiotics to date. As a snapshot of our pipeline, we really break it down to three key categories. And firstly, by method of administration, IV administration, as into human vein, is for the purposes of UTI or urosepsis. We say urosepsis because 30% of patients who have sepsis, which we talk about in just a moment, have an underlying urinary tract infection. And we've demonstrated our compound to be able to go through the human blood, which it carries its desired efficacy properties, pass down through the kidneys, into the urinary tract, and then into the human bladder. We've taken that compound when administered to humans from the bladder, and we've put it out against the offensive bacteria and shown that it is efficacious, and of course, it is safe. We then look to topical applications, so in a parallel program as a gel. So under the term ABSSI, or acute bacterial skin structure infections, our compound is rubbed onto the surface area of the wound. It's got tissue penetration abilities, as in it gets down into the tissue. In fact, we show instances where it gets down into the bone, and it completely eradicates the bacterial infection. ABSSI is everything topical: post-operative wound therapies, diabetic foot ulcer infections, simple skin structure infections. It's a massive, massive market. We recently announced the US Department of Defense awarded a 2 million US, which is approximately 3 million Australian, burn wound infection grant. So we have a US Department of Defense, Department of Army to be exact, burn wound program, tracking nicely as we speak. So what's this thing sepsis we keep talking about? Sepsis is bacterial blood poisoning. It is the #1 most expensive condition treated to date in health. It is double the second most expensive. It has a large patient population of 50 million people worldwide each year, which results in a 1 in 5 death rate, or 11 million people succumbing to that bacterial blood infection. There are no drug therapies specifically approved for the treatment of sepsis. And that same positioning of unmet medical needs goes for our entire portfolio, unmet medical needs of large patient populations. The challenge with sepsis is, of course, for every hour left untreated, the likelihood of survival decreases by 8% each hour. So you present to a clinician, the clinician takes one look at you and says, buddy, looks like you've got an infection going on. And they do two things. They do a blood draw to work out what type of bacteria is in your blood, and if there is an antibiotic that is susceptible to that bacteria. And increasingly, we know the antibiotics no longer work. We, Recce, don't care what type of bacteria that patient has. There is no such thing as good bacteria in your blood. There is no such thing as good bacteria in your bladder. We work faster than any other antibiotic known to date, and we work broader than any other antibiotic known to date. So we're designed to be administered quickly at first patient presentation to stop that infection in its tracks. You can do time-consuming diagnostics. You can work out what type of bacteria it is for whatever reason you like, but we want to stop that infection in its tracks. Don't wait the 24 hours to try to find out, which of course leads to better patient outcomes and reduces the pharmacoeconomic burden of the health system. So how does it work? So some like the techie stuff, some find it a bit overwhelming, but keeping it brief, we basically enter the cell wall. So we enter through small channels in the bacterial cell. And we do two things. We shut down the ATP synthesis, or we shut down the beating heart of the bacterial cell. We shut it down faster than any other antibiotic known to date. We shut it down irreversibly. The bacteria cannot be revived. It works in both actively growing bacterial cells and stagnant growing bacterial cells, or stagnant cells, things like your tuberculosis cells. Another example of our mechanism of action is we stop the Z-ring complex, or that cell division complex where the cells split and divide and infection spreads. We stop that, and we stop that irreversibly. And we have that same mechanism across all bacterial species. If this video works, which it looks like it is, you can see an example at a cell level. Clearly, I quite like pictures, and this is just a great example of how the compound works at a molecular level. Those little guys, the little cells you see there are deadly E. coli cells. So in the left-hand plate, they're growing and multiplying, having a good time as bacterial cells do. Those big guys you see in the middle there, almost like egg shapes, they are your healthy eukaryotic cells. They are the ones you actually want in your blood and body. They are the ones that distribute health and prosperity for the human. Importantly, when we put our compound in the same dish at the same time, the enemy, the E. coli, is dead. It's not growing. In fact, if you're a little adventurous, you might focus upon one and even see it disappear. But the ally, the one that you don't want to target and we intentionally miss, the healthy cell, continues to grow rapidly and multiply, unencumbered, uninhibited, without knowledge that Recce is even present at that time. A really desired targeted mechanism of attack. This is the totally opposite gram stain. So this is a staph infection. Staph, just by way of ecology, don't grow as quickly, so they're not visually as exciting. But you can still see those little circular staph cells growing and multiplying in the left-hand side, and the healthy cells growing and multiplying as would be desired. But in the right-hand side, the really desired characteristic is those E. coli cells really aren't growing, whereas those healthy eukaryotic cells continue to grow and multiply. A desired mechanism of action, targeting bacterial cells whilst avoiding the healthy human cells. So, One of the first studies actually that I sponsored was the ability to show that a compound, aka our compound, can work and keep on working with repeated use. So we looked to the industry side of a protocol that shows that does a compound today work against a multiple different species and over multiple different exposures. So we got amoxicillin by example. They sell at about 10 billion US dollars per annum. After 2 cycles against E. coli, they stop working. That's both negative in the patient context, but also pretty pathetic in product value context. How could you charge a lot for something that doesn't really work? 6 cycles Pseudomonas aeruginosa, 8 cycles staph. Our compound works as well the first time, second time, third time, fourth time, all the way through to a minimum of 25 repeats in all examples tested against. Literature said 25 repeat cycles is the one the industry looks to. We achieved that. We've since taken it out to hundreds and hundreds of cycles and dozens of different bacterial species in doing those cycles. And we always work and keep on working with repeated use, which is not only fundamental to having a capability against the problem, but fundamental to having a value in the commercial marketplace. This is looking at it from the terminology of escape pathogens. So for those who don't know, escape is the acronym that really represents about 80% of deadly bacterial species. Looking across each of them, a 6-log reduction in bacterial count is impressive, but what's even more impressive is it happens in as little as 1 hour. 6 logs is 99.9999% reduction in bacteria. Moreover, those concentrations are practical concentrations that we have dosed humans with in a safe and well-tolerated manner. So broad spectrum capability against multiple different bacterial species. We keep expressing that we work faster than any other antibiotic known to date. And here's an example of that. So here's your more common antibiotics, your cephaloxime, your ciprofloxacin, colostin, that's pretty much poison, tetracycline. And what you're seeing in their therapeutic concentrations when tested against E. coli, the bacteria are still growing, they're still multiplying, the infection is still rapidly out of control. But in our case, it is immediately effective, rapidly declining bacterial viability, which continues to decline down towards zero. It's true that the other antibiotics there would work, but they do work over a long time, really in a lot of ways by method of saturation. It'd take a day, a couple of days to start to see that bacterial colonization reduce. In some cases, it would work in totality. In many other cases, it simply does not. And we've got plenty of special access scheme use examples of that. What's important there, of course, is where you have a compound that works very, very quickly, but has a very short half-life, you have a compound that can quickly bring the bacterial depth count down to really below limit of quantification, but one that's not around for the body to have an inflammatory response to. Where you have foreign matter in the body, a foreign antibiotic, such as those mentioned there, the body is going to try to attack it, assuming it has an immune system to attack with. We don't get an inflammatory response. We don't get an immune system response. Our compound works and then is no longer present. And that's an incredibly desired characteristic. So here's a summary of our first lot of phase 1 IV dosing data. The data demonstrated that up to and including 6,000 milligrams over a 1-hour infusion is a safe and well-tolerated. There were no serious adverse effects. There was no significant changes in any laboratory tests, EKG or telemetry. And we really demonstrated that our half-life, say 3 to 5 hours at higher doses. Yeah, really, it's about an hour and a half, but we do demonstrate, of course, at the higher doses up to 3 to 5 hours. But what we didn't expect to see and very positively saw was the urine concentrations were up to 20 times higher than the concentrations in the blood. Now, what that really does is it lends itself to the fact that if you administer through human vein, through the blood. It passes through the kidney, goes down through the urinary tract system and into the human bladder, where it has an incredibly high concentration of Recce drug. We've shown, of course, here is the human bladder and the plasma, that concentration, when brought across to a urinary tract setting, has the potential to treat UTIs, the underlying cause of 30% of septic infections, through to kidney infections, through to the all-out septic infection. We have the potential to be part of that patient treatment model from the early stage all the way to the advanced septic stage. Of course, does the compound work in the presence of human urine? Absolutely. You got near on a 6-log reduction in minutes, literally minutes, in this example. We took that data and went across to a fast infusion study. So why did we do that? One looks to an hour infusion setting, which we had demonstrated, and you're really looking to people in a hospital setting. What if we could be the setting of first patient presentation, whether it be in the emergency ward or more commonly in the GP or medical center setting? What if we could administer fast in, say, 15, 20 minutes, 30 minutes, and get on top of the infection just as quickly? What that would mean is you could stop the infection in its tracks. You could treat the underlying UTI, the reoccurring UTI or the complicated UTI in a traditional medical setting, which is much more prevalent in the community. And of course, you could limit the number of patients that go through to that hospital setting, leading to better pharmacoeconomic outcomes. I don't know if we talk about it in this presentation, but we have demonstrated that we were very successful in that study. So we've talked a lot about IV dosing. We now start to talk about topical dosing. So we've done 2 topical studies, phase 2 studies. We've done 1 study in topical burn wound infections, and we say topical, what we're talking about there is either a gel or a spray onto the surface wound itself. And we demonstrated, of course, our compound to be safe, but most interestingly, we demonstrated our compound to be efficacious or effective against a broad spectrum range of bacteria. We then took that compound across to diabetic foot ulcer infections, where again, we demonstrated our compound to be incredibly effective against the bacterial species that are prevalent in the ulcerated diabetic foot infection. This is just an example of some of the patients. Patient 1 there, we say escalated therapy. Basically, that 234 kilogram patient had a whole lot of comorbidities. We had to allow them to receive treatment for their comorbidities, which popped up during the testing phase of our compound. Collectively, the patient had a complete clinical response, and that was excellent in that instance. All other patients, infection resolved, infection cured, infection resolved, infection cured. In fact, even patient 4 there, their infection was resolved and they were cured in half the protocol time. So it became unethical to continue dosing something that was no longer there. So they were released early. We've taken the burn wound, we've taken really the topical dosing infection activity that we do and put it into a broad category, a category that captures all. So we vend in the initial data and we then look to everything complicated skin structure infections, diabetic foot ulcer infections, post-operative wound infections, complicated skin structure infections. And when we open up to the FDA recognized space of AB, SSSI, you're looking at a global market size of about $7.3 billion in 2018, which is projected to carry through to about $26 billion in 2032. Interestingly, that represents a CAGR of around, or a compound annual growth rate of around 9.5% per annum between 2019 and or over that time period. And that really begs the question of how does that happen? There's two ways it could happen. 1, the population is growing at 10% per annum globally. I don't think so. Or 2, the current antibiotics are becoming so much less effective in their treatment that the patients are having to resort to, of course, or having a bacterial infection take place because the antibiotics were not able to protect them. I think that is much more likely the case And therefore, this 9.5%, 10% annualized CAGR is a major growth example of the antibiotics no longer working and the need for new antibiotics to become involved. I should warn the following slides. This one's the least confronting slide of the lot, but the following slides, for those who have had lunch and wish to keep their lunch, I may not be conducive with such. So these are examples not from the clinical study itself, but from just special access scheme use to date. Some will breeze through because they're pretty nasty, but this is the least nasty of the lot. This is what's called a diabetic foot ulcer infection. So on the left there, you see clearly a ulcer that has become infected. This patient was unresponsive to 4 times daily cephalexin, which is really like chemotherapy for bacterial wounds. It's very, very strong stuff. Here you can see using our gel just rubbed onto the surface wound itself, that after only 1 day's treatment, the redness is gone, the pussiousness is gone. We told the patient not to pick the scab off. They did. That doesn't matter. We don't create holes. They just decided to pick their scab off. But clearly, after 30 days, you see a completely healed wound. Now, we focus upon the infection. We do not guarantee wound healing. We do not guarantee wound closure. And the reason we don't do that is typically the reason the ulcer presented in the first case is because the patient has lost all skin healing properties. Generally, there's vasculature closure in the area and all sorts of other problems going on. In this instance, where there's this hole that clearly doesn't have any bacterial present, you would put a graft on, you would suture it close, you would do whatever you normally do to heal a patient. None of those things can occur if a bacterial infection takes place. So your platelet therapies and similar would then be viable at this stage. This is a big nasty one. Clearly totally unresponsive to all existing treatments. We came along after 7 days of just gel, so nothing invasive. Clearly the compound does get a little invasive because of its tissue penetrability. But after 14 days of once daily topical gel application, you've got just a big nasty hole in their foot. Now, that is a good thing because, of course, that's where your surgical intervention, where your graft activity and similar can take place. Again, this patient had lost all wound healing properties, but we gave them the chance of avoiding an amputation, which that patient was otherwise going to get, to have a skin closure event. This is an ankle surgery, in fact, that went wrong. So it was an ankle fusion. This is both a gram-positive and a gram-negative bacilli. That's the technical term for a flesh-eating bacteria. So you can see at day pre-treatment, seriously nasty stuff going on there. But after 21 days of treatment, the patient has been completely cured of the infection. So the area is still pretty red raw just because of the extraordinary treatment that ankle area had been under and the effect of the residing effect, of course, of flesh-eating bacteria. But in all the swab counts, there's 0 bacteria identified. And of course, you can see that dried up and in the advanced stages of healing. This is actually a prosthesis. So if you can imagine in the center of a femur, the center of a thigh bone, screwed into that bone itself. This patient is clearly excreting some horrible pus, very, very nasty red infection. But after only 7 days of the gel application being rubbed around that wound area, there's no infection present, there's no pus present. In fact, the wound has dried up and the underlying bone infection has gone from a brittle stage to a fully weight-bearing stage, meaning the patient was actually able to walk home that day. This is the last of the nasties because I know it's a bit confronting, but this is a toe infection, I guess. Osteomyelitis is the technical term. But what's really interesting here is you can see the biofilm. So that's that shininess, the sheen you can see just below the nail itself there. That's actually what bacteria use to protect themselves. And you can see after 7 days of treatment, although the toe still looks a little bit ugly, we can't fix aesthetics, the infection itself beneath is no longer present. The bacterial sheen is gone. What's not shown here is the x-rays of the bone infection is then no longer prevalent, but that patient has been entirely cured of their infection using only a gel. Not invasive at all, extremely effective, very easy to apply, and of course a great patient outcome. Oh, we've got one more. There you go. Very, very nasty on the left. Horrible pusseous ankle fusion. After only 5 days of treatment, there is 0 bacterial infection prevalent. I'll jump ahead. These are some preclinical activities. I'll probably skip those for simplicity. This is our intellectual property portfolio. This area of synthetic polymeric human antibiotics has been a land grab opportunity. We have over 40 granted patents around the world, composition of matter, method of manufacture, curative claims, preventative claims, methods of administration, IV, topical, oral, inhalant, granted in all your major pharmaceutical markets around the world. I mentioned I'm in Indonesia. Why am I in Indonesia? We have a registrational phase 3 study for diabetic foot ulcer infections happening here. DFIs, as it's known, is a major unmet medical need. Indonesia itself has 11% diabetes rate, where you have 11% who get diabetes, 48% of them get a diabetic foot infection, and about 70% of those get infected. Now, if you have an infection, the outcome is dire. If you get your drug approved in Indonesia, you're approved then into the 10 ASEAN countries, which gives you access to a patient population or a population of about 670 million people. We have a memorandum of understanding with a leading Indonesian biotech company, Atana. I'm over here meeting with Atana to extend that memorandum of understanding, perhaps to a more commercial arrangement. Being simple, I'm not allowed to use the otherwise words. We manufacture our compound ourselves. We do it out at our Macquarie Park facility. We don't own the facility, but the people and infrastructure inside are that of Recce. We get 100% yield. We have no expensive waste. We have very low capital cost. It's a few dollars per kilogram of raw material for potentially, on a per kilogram, you'd be in the hundreds of thousands of potential end product coming out. Very easily done, and we do that for all of our phase 1, 2, and 3 clinical studies. World Health Organization recently recognized us as globally the most clinically advanced new class of antibiotic, which that slide touches upon. We recently had a US Department of Defense burn wound grant. We are a leading drug candidate with the US Department of Army, focused upon burn wounds of the frontline fighter. Of course, we would expect in due course this may broaden out to things like veteran affairs and the behind the front line personnel as well. But this is one we announced only a couple of days ago, and we received a very positive evaluation by the independent examiners there. That is Recce Pharmaceuticals, the ASX code RCE. Thank you very much for the opportunity of presenting to you today. Apologies about the stalling in the slides there, but we are getting ahead and we're having a good time doing it. And thank you for your attention.
Great, thanks, James. I might ask people online to submit questions for the Q&A session, which we'll go to right now. I'll start the ball rolling. James, we spent a lot of time looking at the topical applications. You mentioned Indonesia, the registration trial, the phase 2 trial in the acute bacterial skin and skin structure infections in Australia. Yes. And also the US Army grant, which paves the way for further work in topical applications for burns. Maybe we can revert back to the urosepsis clinical trial and maybe talk about next steps for Recce 327 in terms of completion and release of results for that clinical program.
Yeah, so we always are focused upon clinical data. So our clinical data in all instances has been released in its most present form. So for the UTI infection data, by example, we demonstrated that we dosed approximately 25 persons, and we demonstrated that the compound, when it goes through the human body, is then captured from that human's urine and then tested against bacterial species. We demonstrated that our compound is incredibly active. It retains its activity, which means it retains its efficacy ability that if that patient was very sick, would highly likely have been cured had that infection been present. What that means is we now have that data of certainty, or really a layup to success, that when we do next dose, IV, those very unwell UTI uroseptic patients, that we would expect to see a significantly efficacious outcome. We'd expect that phase 2B study to be happening around Q3 of this year, and we would expect that to run for approximately 12 months across approximately 30 volunteers. So we're in very advanced stages. We basically take the data from the first series of studies, package it up, and submit it to the regulator, and that's exactly what we've done.
The other thing, James, you mentioned successful capital raising, congratulations, plus the grant from the US, 18 and a half. So with the grant now, that should be over 20 million in cash, thereabouts. What kind of runway does that give you? And is there any need or any point where you'd be looking for a development partner for any of the programs?
Look, we always look to a development pathway. In fact, our phase 3 study is a registrational phase 3 study. We have expedited regulatory review from the Indonesian FDA equivalent here for that. So that study we would expect to take approximately 18 months, approximately 300 persons, 100 placebo, 200 active compound, and we've got. I'm hesitant saying it because, of course, I'm trying to remember whether we've announced it, but theoretically, many hospitals all lined up to dose those patients. So the probability of hitting those targets is very real in this major patient population of Indonesia. To the cash question, we did a capital raise where we had approximately I don't know, $5-8 million cash at bank. We raised approximately $10 million. That takes us through to about $18 million. We then got approximately $3 million of grant, and then we have our R&D rebate rapidly coming around down the mountain, of course, as we've completed the financial year we're in, and we go into next financial year itself. So we have a monthly burn rate of about 1.6 million per month. That sees an annualized expenditure of about $21-ish million. I'm going around numbers here, guys, but close enough. Which therefore means when you add up how much cash we have available versus how much cash we spend, we believe we're funded into FY of 26.
And you mentioned the pharmaceutical company in Indonesia.
Yeah.
Could they play a part in the development or the end stage of the development and the launch?
Look, I think you really look to a licensing, marketing, and distribution partner. When we say development, the inventive step is long since done. We're really just slap it on the wound, take the bacterial colonization of before, how high was it, after, has it reduced, which would be a sign of hitting its primary endpoint, put that to the regulator, which is a registrational perspective, and then sell. So really, Atana are a licensing, marketing, and distribution entity, a big entity, thousands of staff, in fact. And we're positioned with them with this registrational study right next to the potential of market approval, which is commercialization, and that is sell. And that is what PT Atana is about.
Obviously, being included in the World Health Organization's list is fantastic exposure for the company and the compound. What does that mean in terms of attracting partners in other geographies?
Well, very well said. In fact, it was the central piece to why Indonesia came knocking. We've been working with World Health Organization for some years. The pipeline that they announced maybe a month ago was really actually finalized about 12 months ago, but they take a long time to release these things. So the government, Indonesian government, that is, knew that clinically in that pipeline, we are the most advanced, most modern, most potentially valuable new class of antibiotic known to date. Politically, that is incredibly important. So they basically said, look, we have 270 million people in Indonesia. People are wanting to move away from traditional kind of herbal sort of medicine to Western medicine, to bring themselves up in prosperity, to lift GDP, and to have access to the most novel new medicine for the population of Indonesia. So they said, we want to have at the first access. We want to be seen to be supporting our voting population of Indonesia. We want you, Recce, to come to Indonesia so that we can provide this and be seen to be doing the right thing for our people. So you can actually see the Indonesian Minister of Health there, Minister of Budi. That quote is an approved quote, which I think he basically says something nice and about the challenge of antimicrobial resistance, and Recce's positioning to support that. So we're working hand in hand with the Indonesian government. The huge majority of everything we do here is sponsored by the Indonesian government as an expression of them trying to get the compound for access to their people.
Maybe we can talk a little bit about the pipeline over the next few years, and also the potential for an IND in the short term, and what exactly that will mean for the indications being pursued in the context of the pipeline.
Most definitely. So when we look to our pipeline, we really look to this slide here. We talked about Indonesia mostly just because I'm here right now and it's an active piece, but really I mean I'm in the US next week as well. The crux of the matter is we are US focused, we are IND focused. You've got about 48% of the global antibiotic market in the US, 3% in Australia, 20-ish% in Europe, and then there's the rest. So Indonesia is running in parallel to our US focus. If we look to our programs, we've completed really the phase 1, phase 2A for IV dosing. We're packaging that data up and doing a 2B for UTI or uroseptic patients, where we expect to start that in Q3 of this year. For the topical application, we've announced and we're actively getting ahead with our AB-SSSI study. That study will run for approximately 12 months. We would expect patients, well, more patients than we've already announced, to be dosed in the very near term, and that would position ourselves in both the top instance and the bottom instance to submit an IND application to the US FDA, one for topical, one for oral, both around November of this year. And we've done a lot of work to do that. It sounds like a simple filing. It's actually one of the biggest, most challenging things we've done, but I think the outcome will be very positive. And of course, in the IV setting there, we have an expedited regulatory review, part of that QIDP designation. The burn wound program, that is one that is active with the US Department of Defense at this moment. It's a little less easy for us to have insight and exposure as to everything that's going on, to have such active news flow as we would have and will have in the other program instances, but nevertheless, I would expect significant news flow there. So in essence, yes, we are definitely FDA focused. Yes, we take a portfolio approach. So the upside opportunity of multiple data points, protecting the downside opportunity of having a miss, but we are certainly getting ahead.
James, given that this is a brand new class of, albeit synthetic anti-infectives, where are the challenges? And this is a double-barrel question. How important is the strength of your board, namely people like Dr. Alan Dunton, in terms of addressing those challenges when introducing the product in the US?
I think the challenge of being new and different in any industry is the greatest challenge of the lot, because you're convincing a regulator that something they've never seen before, don't know anything about, to quickly go through that knowledge gap to determine what your thing is and how that fits their regulatory mold, because there are certain tick box exercises that they go through. Thankfully, members of our board have been, Dr. Alan Dunton's an example, but really many or most members of the board have commercialized a therapeutic in their commercial existence previously. But it's really a status of arguing the complexity of the compound, because if this was easy, anyone else would do it. But it is an incredibly complex compound with thousands of known molecules in it, that those molecules have certain characteristics or they don't have certain characteristics, and that they're always in the same position at every single time, and is therefore an arguable intellectual proposition that can be easily demonstrated. We think we have demonstrated that. So by example, that really comes down the CMC path, chemistry, manufacture, and controls. That's always the greatest risk of anything new. But I think we have that in very good handle with a very persuasive argument and the best people to argue that to the regulator.
Great. That brings us to the key milestones we should be looking for over the next 12 to 24 months. Now, you mentioned the IND submission,
Yep.
topical and oral this year. What else
Yep.
should be in the near term investors be looking out for?
Look, this is the year of efficacy. So I see that we will have first diabetic DFI registrational phase 3 patient dosing in around September. In fact, it's currently positioned at approximately the 3rd week of September. And that would be dosing of diabetic foot patients up here in Indonesia. We would expect preliminary data readouts either in cohorts or in whatever capacity possible through the life of that study, which is positioned for approximately 18 months. So expect news flow there. We would expect to start patients dosing of the IV uroseptic study, of course, in approximately the same time frame of around Q3, and we'd expect data points and activity around that to follow. Of the ABSSSI program, we would expect data points, aka patient dosing to date, records to be made available in the very near months. Hopefully we see the continued efficacy we've seen to date, which has been excellent, but we would expect that. And then, of course, the data points that come from the US Department of Defense burn wound program itself. Outside of those, you've got things like we'll be making the opening address at the World AMR Congress or the World Superbug Congress, positioned as a sign of new hope in the fight against antimicrobial resistance. We'll have a very sizable and welcomed R&D rebate, which normally arrives in around November. And we have a number of preclinical candidates in their advanced preclinical stages, which may become clinical in their own capacity, but certainly there'll be data readouts associated with those programs running forth. And then what there's unlikely to be, unless it's some incredible event-driven, is a capital raise. We're very well funded. We express the funding around that into approximately FY of 26. So it's really go, go, go on the efficacy front and the news flow opportunities that flow from that.
Sounds like an exciting time ahead. Thanks,
Certainly.
James.
Thank you, Chris.
I'll just check online. We've been listening to CEO James Graham from Recce Pharmaceuticals. Thank you very much, James, for a very comprehensive update. Look forward to next steps in the near term.
Thank you,
Thanks
Chris.
again.
Many
Thank you.
thanks.
And thank you to everyone online.
Thank you.
Bye for now.